Thank you, Oliver. So our next speaker and the final speaker in this session is Dr. Jonas Bergquist from Uppsala. Dr. Bergquist is a professor of analytical chemistry and neurochemistry, and he has a specific interest in clinical neuroscience, and I think you'll see that. His title is Biochemistry to Support the Evidence of Neuroinflammatory Involvement in ME/CFS, another topic that I know we'll return to during the conference. So it's a pleasure to introduce Dr. Bergquist.
Thank you so much, Joe. And it's a great pleasure to be here. And I'm very honored to be, I think, the only faraway foreigner actually giving a presentation today. So I will bring you over to a place where it's a bit dark and cold still, but we are improving. And we are going to Uppsala, which is a small city in the north of Stockholm, in the country of Sweden. So what made me come to this place and give a presentation? Well, there are different reasons, and some reasons are in the audience, actually. I will get back to that soon. But one thing that I would like to start with is to announce the very good news that we got, and we actually are making public this week, is that the Open Medicine Foundation has agreed to fund a third research collaboration center at Uppsala, together then with the two collaborative centers here in the United States. So Ron Davis Center at Stanford and Ron Tompkins and Wenchong Chow at Harvard University. So that's a great honor and pleasure for us in Uppsala to have the chance to collaborate around ME/CFS with these great teams in the United States. So then, what makes Uppsala special? Well, I had to think a bit about that. It's a very small place. We are not that many people in Sweden, and in Uppsala we are not that many people either. So it's a very old university. It started in 1477. It's among the top 100 universities in the world, apparently to some scores. It's a very small city. 25% of the whole capital or city population is students, and we have a very integrated research center and university campus. And I think that's one of the keys why we have actually been very successful when it comes to developing methodologies and tools and things to use for measuring things. So we have a close collaboration with the clinics, with the preclinical departments, with the research and technology development departments around the city. We actually have one part of the university on an island in the Baltic Sea, which is actually this Campus Gotland here, which we adopted just a couple of years ago to become part of Uppsala University. We have some famous names that came from Uppsala, and you will probably recognize a few of them. So Olof Rudbeck, for instance, who discovered the lymphatic system. And that evolves a bit to what I'm going to talk a bit about today, the immune system and the nervous system when they collaborate or don't collaborate. Carl von Linné, I think you know, and the Celsius, Ångström, Tobin Bergman, Scheele. And we have 8 Nobel laureates coming from Uppsala, and thinking of that as a small city, a small university, that's rather impressive, I would say. We also have some pretty famous former students that comes out from Uppsala University. And Jöns Jakob Berzelius, or Berzelius, was one of the students, and he made his basic studies in Uppsala. But then when he was supposed to defend his thesis, Uppsala did not approve of that, so he actually moved to Stockholm instead. He became a very famous chemist in the end, and was the father of chemistry to many of us. He discovered 4 elements, among selenium and silicon and thorium and some other elements. And he also gave name to proteins. The protein name came from Jöns Jakob Berzelius. He also was one of the leaders to start the Royal Academy of Science in Stockholm. And as you know, Royal Academy of Science, they hand out the Nobel Prize, and that made Uppsala very upset, I think. And now, you know, the Nobel celebrities are held in Stockholm instead of Uppsala. And, well, it's not so far away to go down to Stockholm, but it's a pity, we think. Anyway, he also wrote the first book in clinical chemistry. He wrote it in Swedish, and he named it Lectures in Animal Chemistry. And since it was printed in Sweden and in Swedish, it did not spread around the world so much. But he actually touched upon a very, very interesting subject to me and to some of you in the audience. And it was the idea of looking into different body fluids. And in this book, he presents the body fluid called, in Swedish, brain water, hjärnvatten. And he actually describes this water very, very delicately and realizes the content of this liquid then that surrounds the central nervous system. And I will teach you some Swedish. Denna vätska är vanligt gulaktig, stundom något stickande i grönt. Har en söt, sliskig smak, saltsmak. So it means it's normally clear liquid, sometimes a bit yellowish, even sometimes green. I think that's not really what we see normally. The taste is a bit salt-sweet. So that's what he used as a detector. Luckily, we have other tools today. And he also realized that if you mix alcohol into this liquid, it actually gets, you get precipitation of proteins in there. So there's protein present in this liquid. So how do we study the brain today in Uppsala? Well, we don't taste the samples normally. We use other tools. And we have been quite successful of developing new methodologies for sampling the central nervous system in alive patients. And that's some of the tricky parts of getting to the central nervous system. Of course, imaging is a fantastic tool. And I will just say that. We haven't mentioned that in the lecture otherwise. But combined with that, we use, for instance, filtration technologies called microdialysis. Some of you know it from animal models, but we use it in intensive care unit as a standard procedure for monitoring traumatic brain injury patients, for instance. It's a small device you put into the tissue, and then you can filter out metabolites or even larger proteins nowadays to look at what's happening in the central nervous system. We work quite a lot with one part of the brain that is actually something we can see, so that's the eye. And we monitor things in the eye, for instance, in aqueous humor, which is a liquid you can get to by sampling the frontal part of the eye here, just on top of the lens, basically. We do isolation of extracellular vesicles. One thing we have been struggling quite a lot with is to get specificity and get enough clean vesicles to do monitoring of biomarkers for certain organs, but that's something we work with. We'll work a lot with cerebrospinal fluid, and I will get back to that in the data sets. We do autosurgery and do microprobing through autosurgery. So when there is a surgery going on through the ear, we actually can get samples collected from brain with a small microprobe that we have developed. We do biopsy material and autopsy material, of course. And then we also do things in blood and dry blood spots, which is, of course, something that is more available than many of the other devices that we have here. So for analysis, normally we do shotgun or targeted proteomics using mass spectrometry, or metabolomics that we already heard very nice presentations about. So I won't dwell into the technology in itself, but very, very advanced separation, multi-dimensional separations connected with very high resolution mass spectrometry, with tandem mass spectrometry as the feature where we do sequencing on all the molecules we get. And that leads to a lot of data, and data handling, as we learn, is a complex thing that we struggle with sometimes. Today, I will just tell you what we are doing right now. So how did we explore ME/CFS? And actually the reason is I like skiing, and there was a neuroimmunological conference in Vail many, many years ago. And Vail is a nice place to do skiing. But also we can meet some people that are very interested in neuroimmunology. And Steve Schutzer is in the audience. He was one of the reasons why ME/CFS came onto my table and into my interest. And I will show you later what we did. So you know that the immune system and the central nervous system is communicating in a very, very delicate way. And in many ways, it's a very good way of controlling what the development of the central nervous system is doing, but also the central nervous system controls the immune system, and the communication is super important. But sometimes things go wrong. So for instance, in autoimmunity, we have problems with that the central nervous system is affected by our own immune system. We have problems with dysregulation in the gut. We have problems with immune targets that are affected by the central nervous system and vice versa. So that makes it very, very intriguing to look at, but it also makes it very complex to understand in great detail. So we're trying to get into this and also look for ME/CFS biomarkers in this area. So that led to this paper. Actually, it started with a collaborative work with Steve, where I collected a lot of healthy individuals in Sweden for cerebrospinal fluid, and then we matched and we monitored the healthy control proteome of cerebrospinal fluid in collaborative work. And we followed that up in 2011 with this paper. It's still one of my most downloaded papers, I have to say. And Ben Nathanson, I don't know if you are actually in the audience. I think you were supposed to come to the meeting, but I hope to see you here anyway. Came with his ME/CFS patients, and we compared them with cerebrospinal fluid patterns from post-treatment Lyme disease patients and with my controls then. And what we could monitor at that time with high or deep proteome technology that has, of course, developed since then was a large set of proteins and potential biomarkers in the cerebrospinal fluid. So what you see on this Venn diagram is just that we have a fairly large overlap here of what is expressed in all the patients, in all the controls. And then we have unique features for the normal individuals, for the chronic patients, chronic fatigue patients, and then we have for the post-treatment Lyme disease patients. And what could we tell from this data? Well, we could actually tell in very short that we have a low-grade inflammation that we can monitor with this technology in the central nervous system in both ME/CFS patients and in post-treatment Lyme disease patients. We have a significant increase of synaptic plasticity and neural migration activation. So that could be something positive sometimes, but it also can be a reflection of a neurodegenerative process that is happening and that we try to get rid of or solve. And then what we could see specifically in the ME/CFS patient group was problems with axonal regeneration and repair. So that was the findings from 2011. What we are now doing, and the data I will present now, is just a follow-up on what we have been working on for quite a while, and it's been quite a lot of methodological and technological development going on for making this possible. This targeted proteomics, where we have selected a number of interesting markers that we would like to follow. And we do it, in this case, not with mass spectrometry, but with immunoassay-based methodology called proximity extension assay, as I will show you. So the technique is a development of a breakout from the University of Uppsala, and a company is called Olink now, and it's partly based in Uppsala. Let's see if I can turn off my sound here. So what it does is it can, from a very small sample volume, less than 1 microliter actually, if it's a protein-rich sample like blood, collect biomarkers of different kinds. And what you use is a pair of antibodies that bind selectively to your target. And on the antibody you have an oligonucleotide that can ligate, and then you can do extension assays of that. If you have 2 binders to a non-target molecule, you won't get the hybridization and you don't get any signal. So then you extend the ligase with DNA polymerase, and then you do PCR, quantitative PCR on those amplifications. And then you get a qPCR readout from the signal. And in each panel we have used, there are 92 protein biomarkers. You run it on a 96-well plate format, so you get the data for your selected panel then. Of course, you need to know what you're looking for, and you need to select the panel. And then you can do multivariate data analysis or principal component analysis and sort your patients based on their fingerprint or protein panel readout. So we have done this in 2 steps. We have done one study design where we had used 24 ME/CFS patients with matching cerebrospinal fluid and plasma samples. We have used 24 fibromyalgia patients, and we have used controls. They are all sex and age matched, and all collected at the same, following the same strict protocol. And we have used QC and QA samples included. And this is supported by Open Medicine Foundation, by the Swedish Patient Organization, Olink has also been contributing with their methodology and helping us improving the methodology, and a collaboration then with the pain clinic in Uppsala and with Kaje Gottfrieds in Göteborg University. And for the first set, we run one panel only for 92 selected neuroinflammatory markers. And that's still a lot of proteins that are supposed to be measured quantitatively. And the readout from this still non-published data is that we found 25 CSF proteins that were significantly different between the 3 different patient and control groups. So between the fibromyalgia, the ME/CFS patients, and the controls. And we could see that there is a general increase of neuroinflammatory markers in both fibromyalgia and the ME/CFS patients. Only 7 plasma proteins were found to be significantly different, which was a bit discouraging. So cerebrospinal fluid is much more rich in specific signal. That's what we think of. And of course, that is a backdraw since it's more invasive to get the cerebrospinal fluid. The second study that we just got the data, actually I got some of the final data this week before I left onto the airport. We have done a screening only using very small cohorts, 7 ME/CFS patients, 7 first attack relapse remission multiple sclerosis patients, and 7 healthy controls. So 21 samples in all, which is a very, very small study. But we selected these patients with great care to be able to use them for a set of validations just for checking through all the panels available. Still 6 age-matched controls and all samples collected following the same procedures. And this is also supported by Open Medicine and the Swedish Patient Organization and Olink, and in collaboration with Kaje Gottfrieds in Göteborg. At this time, we had the possibility to screen through actually 13 different panels. There are all the panels available today, so it's about 1,200 targeted proteins then that could be related to neuroinflammation, cell damage, and repair. And you see the names of the panels. They reveal somewhat what they are containing, not perfectly, but you can understand that some of them are more directed into neuroinflammation, some of them are more into cardiovascular, cell repair mechanism, and so on. About half of them were possible to detect in cerebrospinal fluid in more than 50% of the sample. So we set a cutoff there. But I mean, most of the proteins were actually detectable in some point in some of the material. We can also see that the distribution of the samples are fairly well spread over the different panels. So there's no bias on specific levels. You see that the controls on the ME, the green ME patients and the blue MS patients, they spread similarly across the different panels. And we just selected a few of the significant hits we had, and this is just a preliminary data set now, where we have the controls in pink, I guess, the ME patients in green, and the MS patients in blue. And you see, you have significant differences, and what is fairly easy to see is that the MS patients, they protrude out, they're more significantly affected, and you see more inflammation and so on. But in total, 53 different proteins were significantly regulated between MS, ME, and controls. And you also see an interesting tendency, and we see that in many of the proteins, that ME comes somewhere in between the controls and the MS patients, which is telling us something. But you can also see some examples where the ME patients, they differ quite a lot from both controls and MS patients. So we're now looking into this, and based on these 13 panels, we have selected 43 CSF patients that actually comes out as very interesting for the ME/CFS patients specifically. Generally, we can say they still are increased markers for neuroinflammation, cell damage, and repair, and it sort of tags along with what we found in our early studies. And there are both similarities and differences between the ME/CFS patients and the MS patients, which we expected a bit. So summary of what we are doing in Uppsala right now is that we have been able to see these significantly elevated levels of neuroinflammatory markers, both in ME/CFS and contrast patient groups. Cerebrospinal fluid is our target, and we have used untargeted and targeted proteomics for these approaches. What I don't have time to tell so much about today is the other ongoing studies that we are conducting in Uppsala right now, where we, for instance, look at the disturbed steroid pattern, steroidogenesis, that we can find in the ME/CFS patients. And then we target plasma samples, so more easily to access. The signal that we see in the steroid cascade is related to potentially mitochondrial dysfunction, so I think that is an interesting lead. We also, by the help of Solve ME, set up a new thyroid hormone mechanism method for analyzing all the patterns in the thyroid hormone regulation. And our initial data looks that we find a significant change in the ME/CFS patient when it comes to thyroid hormone regulation. We have also been able to verify and repeat some of the studies that have been done by Carmen Scheibenbogen in Berlin, where we find these significantly elevated autoantibodies against adrenergic and muscarinic receptors. And we find them in the ME/CFS patient plasma. We don't find them in the cerebrospinal fluid, so they are not what we think intrathecally produced antibodies, at least, so that could be good when it comes to potential treatment to remove these antibodies. And then in collaboration with Simon Carding's group in UK, we have done a small metabolomic alteration finding in gut of severe ME/CFS patients. And Daniel Vipond in the audience is a PhD. Well, I can say you're a PhD now, right? He was formerly a PhD student. Now he has actually today got a note that he's approved. Congratulations, Daniel. They compared with household controls, which is very interesting as a control group to follow. So by that, I would like to stress the need of being curious. I think this field of research is driven by curiosity from my point of view and from many of you in the audience also. I think we need to think about what to look at. And Bob presented very nicely. We have the tools to look at certain regions. We have to be curious enough to explore regions where we have not been able to go before. Maybe by having some invasive sampling going on, like cerebrospinal fluid collection. We need to continue to explore. So we need to explore what we can get when it comes to the biomarker panels and biological reasons for this disease, to be able to explore and explain and potentially also come to a treatment in the end. And of course, the methodology. The methodology is developed all the time. Back in the good old days where you had to taste your sample or look at the color of the urine of the patient, it's long gone now, and we have all these fancy equipments, but we also have to use them with great care and with understanding that complexity when it comes to instrumentation and analytical techniques also can lead to false discoveries and problems when it comes to data handling. So have that in mind. So I will end up with just thanking all the contributors to the research, and also very warm thank to Linda Tannenbaum, the CEO of Open Medicine Foundation, who have been tremendous helping us with setting up the research center. So thank you, Linda, very much for that. The Swedish RME patient organization for their support, Solve ME/CFS initiative, Ramsey Award there, and Invest in ME in UK. And our clinical colleagues, Kai Gottfrieds, Per Julin, and Simon Carding in UK, and all the patients and the healthy controls that contributes to the research. And thank you very much for your attention. And I have time for a question. Yes, you.
Very nice talk, thank you. I have a question about, you mentioned that you carefully selected controls for some of your metabolomic study, and that's a theme that seems to be recurrent for all three talks about metabolomics and proteomics approach to identifying differences in ME/CSF. Can you say something about controlling for factors such as being recumbent more time during the day, medication, things like that? Has that been studied?
I mean, we try to get the controls, first of all. Early on, the cerebrospinal fluid controls has normally been patients that have been disclosed as non-patients in the end. They've been collected cerebrospinal fluid from due to something, unexplained headache or something like that. We try to avoid that kind of controls nowadays because we now know that they are not perfect controls. So instead we use clearly healthy individuals that volunteers to do a spinal tap. Medication-wise, always difficult to totally exclude influences of medication. We have very good records in Sweden, so that is helping us to try to avoid bio things. And especially when we do these small cohort tests, we have to be very careful with the inclusion criteria. And then I think also it's good to use contrast patient groups as maybe not controls, but as contrast groups, so we can actually look at differences and also similarities between the different disorders.
Have you or others compared freshly isolated versus frozen cerebrospinal fluid, and do we know whether frozen material will be useful?
Yes, we have actually done quite a lot of that during the years, and the freezing process is very important to consider when you are going for long-time storage of samples. So we are following a very strict protocol when it comes to removing cells, for instance, from the cerebrospinal before freezing, because then when you freeze the sample, you would have lysis of the cells. So that contributes a lot to protein background and so on. And also, if you keep them frozen and not freeze-thaw them until the final analysis, actually they are quite stable in deep freezing, -80 or liquid nitrogen. Fresh sample is, it's unpractical to be able to do fresh sampling all the time. But when we do, for instance, if we have possibility to do ventricle cerebrospinal fluid in patients with a drain tube from the ventricles, then we can also monitor this production of fresh cerebrospinal fluid over time. And we can see that it's fairly stable actually over time. So that's interesting to see. Thank you.
Hello, thank you for the presentation. Exertion changes the symptoms of the patient, but certainly also the metabolomics. So are there ways to control the changing state when you make your sampling, like before and after exertion? So would you think that would be important or possible?
It would be important. It sometimes depends on what sampling we are doing. If we do a less invasive sampling, then definitely. But to do cerebrospinal fluid before and after exertion, it would be super interesting if it would be possible to do. I'm not totally sure how we would do that, though. And also, I mean, it depends on the severity of the patient, of course, what is tolerable. So.
Yes, I'm Janet Flint. I'm a patient with ME/CFS. Thank you for your presentation. It's not my field, so I didn't capture a whole lot of it, but I appreciate the work that you're doing. I was wondering, there's the machines, the magnetic resonance spectrometers. I don't know if I said it right, but as a patient, being poked and prodded, having an invasive implant in my brain, kind of, I'm squeamish, and I think, can't we do something non-invasive to capture it and move the field forward without all the need for maintaining samples and controls, and maybe even in a dynamic way? Is that like too far afield, or is that within the realm of possibility? Because I know they're doing it with brain tumors. Yeah.
And I think there will be presentations later this meeting actually about imaging. And that's absolutely, as I said initially, fantastic way of monitoring things non-invasively and looking at dynamics also. So I think we will have presentations later during the meeting.
But is it close in the time horizon, or are we looking further out?
It's happening right now, actually.
Okay,
so,
great. Thank you.
Thank you.
Quick question, your targeted proteomics. You mentioned that there were, I think I heard you say there was only 1,200 proteins available on panels today, or something like that. That's really just scratching the surface of all the protein chemistry in the body. Can you make a comment about how relevant are those 1,200? Are we missing, thousands through the analytics and,
If we consider that the whole proteoisoform pattern would maybe reveal there will be 1 million different proteins, right? These 1,200 proteins are very, very carefully selected for specific functions. So it's not the experiment is not to try to monitor all proteins or random proteins as we do with untargeted proteomics. These markers are selected from a very, very specific point of view. So that's why we are using these 1,200 markers. They probably will develop into more panels depending on what the requests are.
So is it safe to say that these are what we believe are the most relevant 1,200 proteins for those different panels?
Yes. So far.
As far as we know today?
Yes, exactly.
Thanks.
Okay, thank you.